C'est pas des... C'est une fleur, d'accord.
C'est bien ce que je pense depuis le début.
Risa, we will wait a few minutes that people register to Zoom to start, okay?
Ça ressemble à une fleur de type 3 quand même. Je me demander ce que c'est comme ça. C'est une type 3. La fleur, c'est une type 3. Donc une monocotylédone pour... Je sais pas, il y a 3 fleurs, 3 pétales. Enfin, l'autre, on voit moins bien, mais sur celle de bouche, on voit quand même. Il me semble. Je dirais ça.
Je vois ce que tu veux dire. Justement,
sur les autres, on n'a pas l'air d'avoir ça. C'est vrai.
Ça, j'ai vraiment du mal à organiser.
C'est vrai, j'avais tendance.
Okay, so hi everyone. So it's my pleasure to launch today's seminar. So thanks a lot for joining. So as usual, for those of you who are online, you can report any technical issue in the chat, and you can ask your question via the Q&A tab. And for those who have questions, we can unmute your microphone at the end of the talk so that you can interact directly with the speaker, who is today exceptionally online and has not come to Montpellier. And so with that, I'll give the floor to Pierre-Olivier to introduce today's speaker. Thanks a lot, Pierre-Olivier.
So it's a pleasure to introduce Risa Sargent. Risa Sargent is working on plant community and eco-evolutionary dynamics of plant community and pollinators. If I'm correct, she did a PhD in UBC under the supervision of Celiotto. Is that correct? Yes. After that, you did a postdoc at University Berkeley with David Ackerley. And then you moved to University of Ottawa for an associate professor position. And recently you moved because now you are at the University of British Columbia. You moved to British Columbia in more applied science field. That is all plant-pollinator interaction in the context of food security and pollination services. Just an anecdote. I remember a paper you published in, I think, 2012 about the patterns of color of flowers during the season. I don't know if you pursue this research, but it was funny. Yeah. The floor is yours.
Great, thank you very much, Pierre-Olivier, and I wish I could be in Montpellier, but thank you for accommodating the international time zone and all of that. So today I'm going to speak to you about some research going on in my lab recently to do with pollinators in human altered landscapes. And so I'm just going to introduce why I'm interested in this topic and sort of the base reasons for studying the questions that I'm going to talk about. Then I'm going to talk about some work that my lab did in Ontario, in Eastern Ontario near the border with Quebec, on the impacts of seed treatments that include soil neonicotinoids and ground nesting bees. And then I'm going to talk about a second project that looks at how floral resources are a potential management tool and predictor of bee communities. And then finally, I'm going to just give a bit of a taste of some work that my PhD student, Jens Ulrich, is doing, trying to incorporate detection bias into how we estimate the impacts of habitat management on our bee communities. And I'll just wrap up after that. Okay, so this is a paper from PNAS published in 2016. It uses modeling that's tested with a couple of bee data sets. Of course, one of the issues is that we lack very strong bee data sets over time. But the take-home message from this figure, which shows warmer colors, areas where their model is estimating declines in wild bee abundance across the continental United States, and more cool colors are where their model estimates increases. So you can see the pattern across the US is dominated by declines in bee species abundance. And a bit of an earlier paper here used data sets from Britain and the Netherlands to make similar, or to reach similar conclusions about bees in those two countries, and also syrphid flies. So again, warmer colors indicating where, in this case, a data set, long-range data sets were suggesting that wild bees and syrphid flies, both of which are considered important pollinators in natural and agricultural ecosystems, again, dominated by these warmer colors indicating declines. And so, as I'm sure the audience is aware, there are many interacting threats to insects going on in our modern times. Those include global warming, the suburban development, so changes in how land is used around cities and within cities. Major driver, we think, is agricultural intensification, which is happening, at a very fast pace. And related to that, or maybe one of the critical drivers, is the use of insecticides. So constant changes to the types of insecticides and the manners in which they're applied, and the way and the organisms that they impact. And finally, species introductions. So both introduced plants and introduced bee or pollinator species can create threats to bees and other insects. This is a figure from a paper published in Annual Reviews of Entomology in 2011, and I like this figure because it sort of helps to think about what are the indirect and direct effects of the various drivers on, in this case, wild bee, or well, let's stick with wild bees for this one, population sizes. So it's kind of like a visual model of the various drivers. And in the center, you can see the traits of the species that are clearly important predictors of its abundance. So how far can the average female fly? So of course, many wild bees are solitary, so they don't have workers that we're sort of more familiar with in social bees. Their life history is probably an important predictor, how broad their diet is. So can they survive on many different plants, or are they specialized on one or a few plants? And of course, the genetic variation allowing for adaptation or even rescue. And then there are direct effects within the environment. So this is what I'm going to talk about in the second project that I'll discuss, but these have to do with food or floral abundance, the temporal availability of food, nest site availability, and risks, which is what I'm going to talk about in the next section of the talk. And then these sort of broader, more landscape scale indirect effects, such as the way we manage lands, which can include disturbance, may create opportunities for species invasions or allow them to persist or become worse. And so the first chapter or project that I'm going to talk about has to do with one of these incidental risks, especially in North American agro-ecosystems. So I know the situation in Europe is quite different because neonicotinoids have been regulated very differently there. But in Canada and the United States and Mexico, they're still very heavily used here. And so it creates a potential risk to many non-target organisms. So this is a figure from a paper published in Science in 2014, just showing for seed coats in particular. So the seeds are dusted with a neonicotinoid formulation, and often includes fungicides and other agrochemicals. And we know that a lot of the neonicotinoid leaves the seed almost immediately upon planting. This depends on the soil type and the weather incorporated into the growing plant, which is the point. That's why these seed treatments exist. But you can see here a number of avenues through which indirect or effects can occur, direct or indirect effects, but on non-target organisms. So what do we know about how seed treatments impact wild bees? A lot of our knowledge of impacts of neonicotinoid seed treatments or sprays, of course, neonicotinoids come in different applications. This is a nice work done by Madge Rundloff. It was a comparative study of treated and untreated seeds in agricultural landscapes. I think it was done with alfalfa, which is very frequently visited by bees, wild and honeybees. And so they had this paired design where they planted fields with and without coated seeds. And then they estimated general density of wild bees around the fields, the nesting activity of a cavity nesting bee, Osmia bicornis, colony development of bumblebees, a common bumblebee, Bombus terrestris, and then looked at honeybee colony strength of nearby honeybee colonies. And what they found was fairly striking differences among these treated and untreated fields. In particular, cavity nesters had zero nesting success in their treated fields. They saw much lower wild bee density. They saw differences in bumblebee reproductive success. They didn't actually find many differences in honeybees, which is interesting because a lot of the testing for this is done on honeybees. They're sort of a model for ecotoxicology studies in this field. So, just thinking about this paper and what we can take home from it, it's important to note that wild bees, so there's approximately 20,000 different species of wild bees globally, and they exhibit really dramatic differences in their nesting and sociality. And so this is the life cycle of a typical bumblebee species, so species in the genus Bombus. And it shows the overwintering female hibernating, then emerging in the spring, finding often an unused rodent nest, or sometimes she'll actually take a used rodent nest and kind of sting the inhabitants until they leave, and then starts her colony formation with workers and eventually drones. So contrast this with the vast majority of those 20,000 species, of which approximately 80% are solitary. So a single female is provisioning her eggs alone with pollen, and they tend to be ground nesting. So most bee species have a single female provisioning her own brood underground. And so quite different from the bumblebee, which is often also underground, although as you probably know, they can nest in above ground cavities as well. But this shows 4 different nest structures from different species. This is from a review by Cécile Antoine and Jessica Forrest. And it just shows, so the female digs out these burrows out of often soft sandy soil. Then she creates these pollen provisioning balls and lays her eggs on the balls, continuing to provision for several weeks usually. Then she allows the development to kind of continue underground, and bees usually overwinter either as pupae or sometimes as immature adults, or just kind of subadults. But almost everything we know about the impacts of neonicotinoid seed treatments and sprays comes from non-ground nesting bees, in spite of the fact that they're clearly so prevalent, or sorry, in spite of the fact that most of our bees are ground nesting. And so this is a meta-analysis published last year in Ecology Letters, showing from all of the studies that were available to be tabulated together, that there are strong effects on reproductive output. But you can see here that all the species were in these 2 genera. So Osmia is a cavity nesting bee. It nests above ground. And as I pointed out, the bumblebee species have this using existing burrows life history. There is one really nice set of work. This is by Susan Willis-Chan and Nigel Rain at the University of Guelph. They have worked with squash bees. This is a native North American species, species in the genus Peponapus. And they're very closely associated with squash, both wild and domesticated squash. And they did a test. So it's a nice system because the bees are so specialized on squash. You can see the picture here. The bees form their nests usually right next to the squash plants. And so they use this system to sort of do a tenting type design and test a number of neonicotinoid formulations on reproductive success in squash nesting bees near their treated plants. And you can see from the graph here, they find strong differences mostly from soil drench applications. So this is where they just pour neonicotinoids around the base of the plant, but they do find impacts, at least in 1 year, on offspring in the seed treated system. So suggesting that maybe the effects aren't quite as dire, at least in this system from seed treatments, but still some effect on offspring production. And so just to sort of narrow in on how this is happening. So there's a number of routes by which bees could be exposed to agricultural pesticides, including neonicotinoids. So they could be directly harmed during spraying or through the dusting when seeds are planted. If they're dusted with neonicotinoids, they can kind of become airborne. But there are also indirect effects, as that science paper figure showed, via soil. Bees often will quench their thirst or acquire their needed water from standing water. So puddles can be can have neonicotinoids or other agricultural pesticides. And then, of course, the point of these seed treatments is to embed the tissues of the growing plant with the pesticide, and that includes nectar and pollen. And so with the first project that I'm going to discuss, what we were interested in was whether the life history, so ground nesting or not, impacted the impact risk in a field setting. So hardly any studies have been done in field settings, most are in lab. So this was a graduate student of mine, Emma Gaudreau. This was her thesis work. I also had 2 collaborators from Environment Canada and Agriculture Canada, David Lapin and Alona Najakatidis. So because the use of neonicotinoids in Canada and Europe is so different, I'll just kind of cover what the differences are. So the most common use of seed treatments is in corn and soybean seed for purchase. It's actually very difficult for growers to acquire non-coated seeds. So and this is one of the most common crops in Canada and of course in the US Midwest. And so most growers are planting these coated seeds mainly because they can't get anything else from their growers associations. They're a powerful insect neurotoxin, so they're not discriminating among insects. They're not specialized for one group or the other. They interrupt neurotransmitter function. So basically the insect starts to twitch to death, or it has other impacts, changes foraging behavior and cognition in lab studies. There's very few field studies. So that paper I just showed you with the squash bees is one. There's another sort of survey study out of Iowa. Sorry, it's very early here. I didn't think I would have noise in the background. And so just to point out that corn and soybean are some of the most commonly grown field crops in Eastern Canada and the US. And then on top of this, seed treatments get sort of a loophole in the regulatory framework in North America. In both Canada and the US, they're not considered pesticides. So they're not regulated as pesticides and they're not treated in the same way. So neonicotinoid sprays are more heavily regulated. This is something that policymakers are working to change, but it's taking time. So our study was done in corn and soy fields in Eastern Ontario. You can see the map of Canada here on the left side. So around the Ottawa region, and you can see the blow up here. So the major cities, or they're very tiny towns kind of around the Ottawa-Gatineau area. And we approached farmers to have access to their land and permission to do our study. And so after sort of, doing lots of requests of various farming communities, we ended up with 18 fields to study. The common growing pattern in this part of Eastern Ontario is to rotate between soy and corn. Occasionally, those will also have a hay rotation. So we ended up with 18 fields that were in this kind of rotation. 4 of the fields were organic growers, and 13 were conventional. And because of the difficulty of finding non-treated seeds, we suspect that all of those conventional growers were growing coated seeds. We tried to control the size of the fields. All fields had to have a hedgerow, so a kind of unmanaged weedy border among fields. And we wanted them to have spatial independence, so they had to be a minimum of 1 kilometer apart. Some of them were up to 5 kilometers in distance from each other. So then we performed bee surveys over each month in May, June, and July, August 2019. This was a 2-year study, but we ended up focusing on the 2nd year because of difficulties we had just kind of getting the full 4 months of surveys in the first year. We then identified bees to all bees to species where possible. We took soil samples from these fields within about a week of when we could detect the crop was going in, so soy or corn. We also measured, you can see the hedgerow here. This is one of our fields. We used quadrats to get some idea of what the floral resources were in these hedgerows, because that's expected to be a strong predictor of bee abundance and diversity. And then we used generalized linear mixed models to look at marginal effects of the detected soil neonicotinoids, also the type of crop and the floral resources on both abundance and diversity of not just ground nesting bees, but all bees in the ecosystem. For the soil pesticide testing, we farmed this out to a lab at Agri-Foods and Agriculture Canada. Tests were run to establish concentrations of the 3 major neonicotinoids that are known to be in seed treatment. So these are imidacloprid, thiamethoxam, and clothianidin. The soil samples also were tested for a suite of other agrochemicals at the same time. And then because one part of our idea was that ground nesting bees may be reacting differently. They may be, because they have additional exposure routes compared to, say, cavity nesters or bumblebees, which don't, ingest and dig out their own burrows typically. We also classified every species that we caught as either ground nesting or not ground nesting, using taxonomic descriptions from the major bee literature. The most common species that we found in our data set were bumblebees in the genus Bombus. As I've already mentioned, they don't tend to dig their own holes, so we classified them as non-ground nesting. The data was then divided into these 2 classes, and we ran the mixed models separately to see whether we could detect differences among the 2 groups. So this is the soil neonicotinoid data. We only really detected clothianidin and thiamethoxam in any significant concentration. We didn't detect imidacloprid at all. Our best estimates of neonicotinoids in the soil came from the crop soils. We also had estimates from the hedgerow soils, but those were done at a different lab, and the concentration, the detection levels were higher. And so we just scored them as present or absent. And we then binned our fields into 3 categories. So some had no neonicotinoids detected at all. Others had high levels, so those were with above 4 parts per billion. And sorry, yes, and then we had a low, which was between higher than 0 but less than 4. So you can see here the data fell fairly nicely into these 3 categories. And so what did we find from these models? First of all, we found that, not surprisingly, that hedgerow floral resources were a strong predictor of the bee abundance detected in foraging in those hedgerows. We also found differences among, in terms of the abundance of all bees among our categories of high and then together low and no detection were not distinctly different from each other, but the higher level neonicotinoid fields tended to have a lower abundance of bees. We also looked at species richness of the bees, and this followed a similar pattern. So floral resources were also a predictor of bee richness in these fields, as were neonicotinoid field categories. So the high, again, had fewer species and fewer bees overall relative to low and no detection fields. We didn't find any differences among bees according to nesting habit, though. So that was one of our predictions, was that we might see stronger effects in ground nesting bees because of their additional route of exposure. We didn't find any discernible differences in these patterns. Ground nesting bees and non-ground nesting bees followed very similar trends. It's possible that, you know, it's a one summer set of data, so it's possible that we just don't have enough power to detect any differences. It's also possible that the effects are relatively weak and don't, you know, or maybe the effects are largely through these plant products that, you know, pollen and nectar exposure, and soil exposure doesn't have as much impact as we thought. Most of our non-ground nesters were bumblebees. So they do nest in the ground, but they don't dig out their burrows. So it could be that that's too subtle of a difference to detect any differences between the groups. So not totally clear why we couldn't support that hypothesis, but that's what we found. So our findings are consistent with the Willis-Chan data showing differences, at least in reproduction, which should bear out in abundance over time. They're also consistent with a very similar study that was published kind of as we were basically working up our data in 2020. So fields in Iowa with and without seed treatments, and they also found differences mostly in abundance. I don't think they found diversity differences among their fields. So it seems quite likely, based on this data, the meta-analysis, the Madge-Rundloff study, that wild bees are facing critical hazards in environments where neonicotinoids are present in the soil and presumably in sprays as well. And so one of the clear implications of this is that the regulatory framework that's applied in North America should come into line with these hazards to non-target organisms. So the second part of the study, the next set of data I'm going to talk about, is a meta-analysis that was done with my PhD student, Batul Hijazi, and this was to compile evidence across as large a set of data as we could about how floral communities could predict bee communities. And so this is back to this figure from the Ralston and Goodall annual reviews paper, just showing that we're kind of moving around their figure and looking at a different potential direct effect of bee population size. So, so one thing that's critical to know in order to kind of understand the theory behind this, this part of the talk is that bees are what we call central place foragers. So from the center of this image here, you can see two types of bee nests. So the left is cavity nesters. So somewhere, this is obviously a man-made one, but they basically use little cavities that they find in above ground structures. And then on the right, this is an example of what you would see if you detected a ground nesting bee nest. So you can see it sort of looks like an anthill, the little hole, and you would see the female kind of coming to and from the hole. And so most solitary bees, which as I've pointed out, are the vast majority of our species of bees, are foraging within probably 150 meters of their nest. So obviously this, this distribution has a long tail, and it's probably correlated with the size of the bee. So smaller bees probably use a smaller range of foraging. But what that means is that the resources directly surrounding the nest should be strongly predictive of the ability of the bee to survive and reproduce in this area, or one of the predictors. And so that's kind of the basis of this study, the theoretical basis. Excuse me. We talk a lot about different pollinators. I focus on bees quite a bit. And one of the reasons bees are suspected to be such important pollinators in both wild and agricultural systems is that they're the only pollinator, or one of the only pollinators, that relies entirely on nectar and pollen, probably within about a 500-meter radius, to rear their offspring. And so because of this, these local resources, if you could measure them close to the nesting site, should be strongly predictive of bee abundance. And then for a variety of reasons, we also suspected that community diversity should predict the diversity of bees. So this includes reasons such as specialists will only be included in diverse communities that include their specialized food source. Diversity could also allow a longer seasonality of bees. So if we have a number of species that kind of flower throughout the season, that may allow bees with different phenology. So again, richness potentially predicting diversity. So what Batul did was she did a defined Web of Science search for papers that examined relationships between floral resources and various ways of measuring bee communities, mostly abundance and diversity. She identified a total of 534 species, but after careful review to sort of sort through which ones were relevant for the question, sorry, I can't see my Zoom is covering the number there, but I think it's 60 something. So a total of 133 effect sizes. We then used meta regression to examine the overall relationship between floral resources and the bee community. And we also looked at specific relationships, just curious, like which, if we could tease apart specific drivers of bee abundance and diversity from the plant community. And finally, our data set included both enhancements, so purposefully planted plant communities, as well as existing floral communities. And so we were interested to see if there were detectable differences between these kind of management style or human planted enhancements versus what had been existing for many decades or more. And so this is just a picture of what we mean here. So you can see on the left, this is a planted hedgerow in California. You can see the deliberate planting of specific species right along the margin of the field there. And on the right, this is actually from Emma's study. So these weren't, some of the plants could be deliberately there, like a farmer may have put specific trees as wind breaks, et cetera. But a lot of the community is just kind of naturally assembled there from the native or whatever is available in that region. So most of the effects that we gathered from these studies were from agroecosystems. This is a common thing to do in agricultural studies, or agroecology, is measure the plant community and related pollinator or bee community. Many of these were from orchards, some of them were from field crops. 15 of our effects were from so-called natural areas. So this included parks, grasslands, this type of habitat. Only 7 were from urban areas, which is interesting because I think this is an area of growing interest, but not a lot has been done in terms of just measuring bees and plant communities in urban areas. Not surprisingly, half of these studies were from Europe and approximately the other half were from the United States. There were a few from South America and Asia. And overall, we found very clear positive relationships among vegetation survey results and the bee community, regardless of whether it was enhancements, vegetation, agroecosystems, urban. This is the results in ordered by the effect size on the left. So I realize you can't see the authors there, but just to show you the range of the effect sizes. And then the summary was an overall correlation of 0.71 between floral resources and bee community measured as diversity or abundance. And so when we broke these down into various types of studies, so some looked at floral abundance and bee abundance alone, some looked at floral abundance and bee diversity, others at floral diversity and bee diversity, and finally floral diversity and bee abundance. We didn't find any significant differences. You can see that the final effect is on the lower side relative to the others. We didn't predict that. We're not really sure why. Of course, there are correlations among all of these, so it's hard to say, what the exact drivers are. But we decided to kind of pull the studies into these categories to see, whether we could see differences in the different associations. But there are no clear ones. So the implication of this is sort of it's a nice compendium showing the overall effect size of floral resources are indeed a strong predictor of the size and diversity of local bee communities. As I already mentioned, most of these are from agroecosystems, suggesting we may lack data in other kinds of ecosystems because this is a very broad literature search. And we didn't find any differences between enhancements and existing plant communities. So that's interesting because it suggests that enhancements can be quite effective, but also, of course, it gives more justification for continued conservation of existing plant communities for bee conservation. And so the last thing I'm going to talk about, this is a photo of a PhD student in my lab. This is Jens Ulrich, and he's interested in estimating effects of these kind of enhancements in urban areas. So you can see the 2 types of habitat here in the photos, Jens and some field help doing vegetation surveys in a heavily managed park. These are all from the city of Vancouver. And he sort of took advantage of a new management, application that started during COVID in the summer of 2020. I think the city just didn't want to send as many people out to mow parklands. And so they decided that they would keep some areas unmowed for that summer, and then they've continued to manage some parklands very differently. And so Jens was interested in how, whether he could see any impacts of this on bee communities in terms of abundance and diversity, but also recruitment. So he's doing a number of different studies to see the effects of these changes in management. One of the things, a lot of these studies are looking at associations between plant communities and bee communities, but it's hard to know whether bees are just moving into the areas and so maybe potentially depleting other areas that also, require bees, or do them, ideally, these management schemes would actually improve population would improve reproduction in these habitats. So not very much has been done on this. It's really unclear for most management schemes whether there's any impact on nesting and reproductive success of bees, or whether we're just kind of moving the existing bees around in the landscape. So Jens is trying to get at that with a number of different studies. So I'll just show you it's kind of a preview of something he's been working on recently. So this is his big question. These are pictures of two parks that would have previously been managed very similarly. So on the left, you can see one of them is mowed every 2 weeks. That was the old scheme. So for the last 40 or 50 years, these same parks have been mowed every 2 weeks. And then starting in 2020, they stopped mowing. And so you can see that the stark difference in the floral abundance, and a big change to the landscape in terms of what's there, most likely impacting pollinators in some way. And so this part of the talk just kind of gets at a little slice of one of the things that he's looking at. And so Jens has identified kind of an assumption of many of these enhancement studies, like the ones that we are pulling out in the meta-analysis that I just showed. Usually there's an assumption that the ability to detect species in the two habitats is the same. So it's kind of an underlying assumption of all these studies. As I pointed out, we almost always see differences among communities with different among habitats with different plant communities. And sometimes that difference is quite it's quite a large effect. But there's this implicit assumption when we're doing these surveys in these two habitats that they estimate the bee community in the same way. So there's no bias with respect to our ability to detect bees in the two habitats. And so Jens is interested in whether that's a reasonable assumption. And so he's identified two possibilities for detection bias. So one is that the habitat itself shifts the ability to detect some species, or maybe all species on average. And so you can see here another picture of these communities. I think Jens's initial hunch was that detection would be easier in the unmowed sites. So more flowers, maybe just easier to see bees when they're on flowers. Maybe that impacted the ability to detect them in these landscapes in some way. So he'd initially suspected that there could be, or he hypothesized that there could be a detection bias in the direction of easier to detect some species, or maybe all species on average, in the enhanced habitats, or the unmowed habitats. The second possibility that he's identified is that it could differ among bees regardless of habitat. So large bees, again, we're using our human eyes and brains to capture these bees. We're doing most of the captures are by net capture. So you see a bee usually foraging on a flower, you capture it, you identify it back at the lab. So it could be that just sizes among bees, or their shapes, their behaviors, something about the species itself is a factor in detectability. So to test this, Jens used a technique that's been worked up for including detection bias in surveys of population abundance. And so over a short period of time, I think he used a week, 8 days. And the reason for this is because one of the assumptions is that the abundance isn't kind of naturally changing. So you're not hitting something in the middle of its phenological cycle and losing it from your sampling anyway. So we know the phenology of these bees quite well, and we know what bees are more common in these landscapes. And so he did repetitive studies, 3 surveys per site, on 7 different species of bees that he knew from his previous collections were existing in all of the 10 sites that he used for this estimation of detection bias. And so he used 5 sites that had this no-mow and 5 that didn't. And then during this short time window, during the predicted peak abundance, he did these repetitive surveys. And so then we modeled the effects of covariates on the abundance and detection probability of the different species and species on average, and estimated model parameters using a Bayesian framework. And so these are just preliminary results. I'll show you in a slide in a minute that Jens would like to repeat this and change some things. But what he found, so this shows these estimates of the detection probability in the two sites, and the slopes are different species, so the 7 different species that he captured. And what you can see is that the average and across the board detection probability was actually higher in the unmowed sites, so the opposite direction from Jens's original hypothesis. And so suggesting that there is detection bias in the system and across species, across common species, and that it's they're more easily detected in the no-mow environments. So for this part of his study, he's going to repeat this next summer and potentially at a larger scale, so with more sites and more observers. He's also interested that this is not a mark recapture, he's simply noting the individuals and counting them. But he's also applied a mark recapture approach, so he hasn't looked at that data yet, but that might be a more, it's more robust to some of the model assumptions that are built into the approach. And he's also interested in maybe looking at, so right now he's looking at very common species, but he's interested in comparing rare or specialized species with potentially the ability to consider effects on interannual population dynamics. But overall, if he does, if he's able to support this finding that detection bias needs to be considered, that's something that should probably be built into methods in the future. So just to sum up what I've shared from our group's work. So neonicotinoid seed coats, not just from our work, but from a growing body of work, seem to pose a serious hazard for bees in agroecosystems. So in Canada and the United States, we're pushing that regulatory changes need to be applied as soon as possible. My student Batul's work showed that plant community enhancements as well as existing communities are critical or a strong predictor of abundant species-rich bee communities. And then this sort of taste of Jens's thesis work suggesting that detection bias is possible among habitats with and without enhancements. And because that's a very common approach in this field, I think hopefully if this continues, he might be able to publish something suggesting that be included in the way that we make these estimates and methods for best practices. We have a number of other things going on in the lab, so I'll just quickly kind of highlight those. So we're together with Lethan McGonigal, who's a faculty member also in Vancouver, but at Simon Fraser University, we're exploring impacts of land use, mostly agricultural land use, on bumblebees using a large North American and European data set that's collected and managed by Leif Richardson. And I have a student who's interested in the impacts of introduced bees on native plant pollinator networks. So he's using a big assemblage of network data to see if he can tease apart differences in the way networks operate. So connectedness, species richness, whether there's invaded or whether there's introduced or non-introduced bees. And I'll just quickly highlight that I'm working with Claire Kremen and Julie Carrillo, who are other professors here at UBC studying impacts of agricultural management and habitat diversity on bees and other insects in agroecosystems. And we actually have a postdoc position that's open right now. So if anyone's looking for a postdoc in this field, send me an email, please. And I'll just wrap up by thanking my funding. It's largely from the Canadian Natural Sciences and Engineering Research Council. I've received funding from the province of Ontario. The city of Vancouver has generously, well, first of all, agreed to continue this management scheme so that we can study it for several years, and provided other sort of in-kind support. And of course, all the students who've helped in the field over the various years who are listed here, some of their photos. So that's the talk, and I'll be happy to take questions.
Hi, can you hear me?
I can,
Okay.
yes.
Thank you for your talk. It was very clear and interesting. There was something that caught my eye right at the beginning when you were talking about coated seeds, and there were several species of bees. And I guess you're working more on wild bees, but I thought I saw that the honeybee was less affected, or maybe you mentioned it too. But I wondered if you have some information about that.
So yeah, it might be from this study here. So this is this Nature paper that looked at several different groups of bees, and they found more pronounced differences among different species of wild bees and very little impacts on honeybees. There could be a number of reasons. Certainly the original concerns about neonicotinoids did come from honeybees. So original applications were like spraying. And so it was beekeepers that started, I think, to highlight the issues with these agrochemicals, because they were seeing die-offs near sprayed fields and were concerned. So, but these are seed coats. So it could be that, as I sort of presented in this chapter, maybe honeybees, because they really have no soil exposure, maybe with the seed coat applications, they're less likely to be impacted. A lot of our information about the impacts of neonicotinoids on bees in lab settings come from honeybees, and it's very clear that they are affected. So if you feed them to them, if you put them in, in their environment in a lab setting, their survival declines, their lifespan declines, they produce fewer, queens produce fewer eggs, etc. So they're not unaffected. It's just this particular study in the field. Obviously, there's lots of other factors going on. So I can't remember what they discuss about the impacts on honeybees. And I think in other systems, they have seen impacts. So it is possible to show impacts, but this particular study didn't.
Okay, thank you.
Oh, I can't hear right now.
Can you hear me now? No?
I can, yes.
So, does it work? No. Okay. At the beginning of your talk, you were talking about the different causes of bee decline. I have the feeling that we tend to put every cause as equal, but do you think there are major causes and secondary causes? This graph.
Yeah, sorry, was there something?
Yeah, this one.
I think we just don't know. I think this is, kind of the holy grail would be to tease these various factors apart and put nice effect sizes on direct and indirect pathways. One of our biggest limitations is data. So very few long-term data sets exist on almost any species. So the one I mentioned at the very end of the talk that's being managed and collected by Leif Richardson is one of the best. It goes back, I think, to the late 1800s, but it's based on museum records. So collectors would go out, they would, come back with, these are all bumblebees, so with a set of bumblebees, but we don't know if it, were they searching out a particular one and only or 5 and only you know what I mean? They're not the kind of surveys you'd want to do if you wanted to have a really nice long-term data set of what was happening in these regions. So we do, it's very nicely detailed in terms of where the samples were taken, but we don't know what the collector was doing. So people are using that with the assumption that we can use it, say, for very coarse types of, I guess, metrics like, did it occur here during this time range? But even that has problems, right? Because a presence means, yes, it was detected, but what does an absence mean? Does it mean the collector caught 500, but they didn't care about that bee on that day in 1895, and so they didn't bring them back to the museum? And that's bumblebees, which I would say is the best records we have over. So really, to know any of this, we need better data. And it's very hard to come by. So we're working with these very patchy data sets and trying to make conclusions, but it's very messy.
Okay.
Risa, thanks for your talk. I was just wondering, in your data set and also in other data sets that where you showed that seed treatment leads to a decrease in pollinator abundances, would you have any data that could tease apart scenarios where the bees are less abundant because they actually die, from a scenario in which actually they disperse to another nearby place where there's not this treatment and this is not occurring? Like, do you have any spatial information on the effect of this treatment on abundances?
No, I don't think that's known either. So whether bees, yeah, I mean, it's possible that we don't know. I mean, this is another really tricky thing, is that studying nesting in ground nesting bees, there's almost no studies of it because it's so hard to find the nests. And yeah, that's the major issue. So it is, yeah, it's hypothetically possible that a female would start a nest and then maybe she detects she doesn't like the taste of the soil or whatever and disperses somewhere else. That's a great question. I don't think we have any idea what information. We're pretty sure they're detecting like substrate consistency. We know that they tend to like soft, sandy soils. Obviously, it's probably easier for them to dig, and maybe the structure holds better in specific soil types. But whether the female is also using additional information, it's hard to say. They're not great dispersers. We don't think, like, many of them probably rarely move more than a few hundred meters, like even over generations. So females emerging from those nests in the spring are probably, in the same landscape. So if you have a landscape like this one in Southern Ontario that's just blanketed with conventional soy and corn that's largely treated, they may not have a lot of kind of escape habitats to go to. But maybe that's what the importance of these hedgerows are. Like, I think a lot of people, like Claire Kremen, for example, is really pushing this kind of inclusion of habitat heterogeneity into agroecosystems for these reasons. But that's a good question. No, there's a lot we don't know, even from, like, I think this is what makes, hopefully, my student Jens's work quite unique, is he's trying to pick apart, like, can we see, using these mark recapture, can we see differences in population dynamics, or what you'd actually want to know, like, are we just watching the same bees moving around to different places, or are we actually seeing the kinds of effects that we'd hope for, which is that these habitats become sources of increased bee abundance and diversity, but we don't know that yet, I don't think.
Thanks.
Okay, any questions? No, on the Zoom? Last question from me. The link between pollinator diversity and plant community diversity, would it be possible that the fact that a resource will be continuous with a higher plant diversity is a big driver of the maintenance of many bee species?
Yeah, that was one of our, we had in this paper, we kind of identified different drivers of the, just looking for the slide there, of the different relationships. So that was one of our ideas, was that more species might spread the flowering resources out over a longer time. We know that bees also use this temporal space, so different, you see a very distinct kind of species phenology of bees. You know, I don't think we know how well it's associated with particular plant communities, except maybe in a few very well studied systems. But that was one of our ideas, was that I guess it would be like a niche space, right? A temporal niche space could potentially drive in plants, could drive or could be associated with a temporal, a larger temporal niche space to be occupied for the associated bees.
Okay, thanks. So if there are no questions, we will close the seminar. Thank you, Risa, again.
Thank you so much. Have a nice weekend.